while. He's a colleague and a friend and a coworker at times. Rich started out of life as a chemist and got his PhD from University of Colorado Boulder, and then went to work at one of the premier foundational laboratories in glycobiology and glycoscience at University of Washington, where he postdoced with Stuart Kornfeld, and then had the bright idea of joining the CCRC for a while. And we had great fun working together, got to know his skills for a while, and he took the opportunity to take a leadership position at the Greenwood Genetics Center, where he's currently the director of research, and has unique opportunities to think about the clinical relevance of mutations that might affect. Well, he'll tell you about it. I don't want to give anything away. But we're really happy to have Rich here. We're hoping that we can have some dialogue and think about how some of his data might incorporate into Glygen and other knowledge bases around the world. And we're looking forward to hearing from you, Rich.
Awesome. Well, thank you both for those generous introductions, and I'm looking forward to taking you through this story. I know some of you saw a good bit of this last week when we were in Georgia for the Georgia Glycoscience Symposium, but I have a few new things to share. And I have a slide at the end where I offer up some ideas and next steps and I guess modes of engagement that I would love to have with this group to expand this project, to enrich it, and we'll just see where that discussion goes. If you have questions, please put them in the chat. If you have a burning question, feel free to break in and stop me. I have no problem with that. But yeah, as Mike said, I really have had over the last probably 20 years an immersion in the fields of glycobiology and lysosomal biology, and since moving to Greenwood, kind of opened up a third front, and that's functional genomics. So I'll talk a little bit about that general effort and then share with you how it kind of brought me back into glyco at the end. Oops, let me see here. Okay. So let me start by framing this, just talking about this challenge in the field of these variants of uncertain significance. And for those of you that aren't in the field of genetics, I'm going to use variants synonymously with mutations. And so I'll talk about this challenge, and then I'll talk about an example where we have tried to fill this gap of understanding the effects of uncertain variants by developing functional platforms, and that'll get us into the lysosomal side of the story. Then I'm going to finish with this story on where it brings it back into glyco, talking about gain-of-glycosylation effects and also into therapy. So yeah, without further ado. The field of genetics, medical genetics, is doing a lot of genomic sequencing. This sequencing generates a lot of variants or mutations, and a large fraction of these variants have an uncertain clinical significance. They've never been seen before. We can't predict what the clinical outcome will be just looking at the genotypes. And so when a provider orders genetic testing and they get a report back and it has multiple variants of uncertain significance or VUSs, it makes the interpretation of the genotype very challenging. In some cases, this can delay decisions on whether or not to start treatment for a patient and just in general how to guide their medical management. So there are a growing number of, obviously in silico-based efforts to resolve uncertain variants. There are other efforts that like multiplex assays of variant effect, where people are trying to look at the effect of every possible variant in a gene with specified assays. But a lot of the work that's being done is really driven through the development of well-controlled functional platforms that you can use to assess variant effects and then hopefully use that information to confirm a diagnosis. So the value of a lot of these platforms is that it goes beyond just functional resolution into beyond interpretation, but you can also use the tools that are generated for the assessment of treatments. And I'm going to talk a little bit about that at the end. But let me give you another example, and it'll also bring in this concept of amenability. So this is just a slide that shows this ad for Galafold. Galafold is a pharmacological chaperone for a lysosomal storage disease called Fabry disease. It's a sugar analog called megalostat. It will bind to misfolded alpha galactosidase. That's the gene or enzyme that is the cause of Fabry disease. It'll bind in its active site and help stabilize and traffic the variant enzyme or chaperone it to the lysosome, thereby increasing its activity. And in this ad, you can see it says for adults with amenable GLA variants. This is what's designed to work for your enzyme. So this chaperone therapy won't work for every GLA variant. It's only going to be the ones that lead to misfolding. So how do you determine which ones are amenable? This group basically developed an assay using a similar approach to what I'm going to talk about. You take a cell line that is missing GLA and you reintroduce mutation-bearing or variant-bearing forms of the enzyme. You express that enzyme and then you measure activity, and you do this in the presence or absence of your chaperone, and you get data that looks like this. So here you can see all of the different mutations or variants that they tested. The activity without the chaperone is in these open bars. Many of these have absolutely no activity, probably because they're misfolding. And then you do it in the presence of the chaperone, and you can appreciate that some of these had no activity without the chaperone, but now all of a sudden they have appreciable activity in the present. So those would be the ones with red arrows are the amenable variants. So I want to introduce that because that comes up in the back half of the talk. But we're not going to talk about Fabry disease. The lysosomal storage that's in focus today is mucopolysaccharidosis 1, type 1. So this is a deficiency in a GAG-degrading enzyme, alpha iduronidase. It's encoded by the gene IDUA. A lot of classic lysosomal storage disease features: enlarged spleen and liver, short stature, skeletal issues, very characteristic coarse facial features, which I think you can appreciate there. A lot of sensory systems like the eye and the hearing are also affected, as well as issues with the heart. For MPS I, the GAGs that accumulate due to loss of lysosomal activity of this enzyme are dermatan and heparan sulfate. Clinically, you can characterize it in three main categories, although it's probably just a spectrum. One is Hurler disease, which is the most severe form, Shea, which is attenuated, and then its intermediate form, Hurler-Shea. There are fortunately approved therapies for MPS I. They include enzyme replacement therapy as well as hematopoietic stem cell transplantation. And because there are therapies, there is newborn screening available for this condition, along with all the other lysosomal disorders that have approved therapies. So it was driven by the availability of therapies that MPS I was added something called the RUSP, but that's the Recommended Uniform Screening Panel, which gives guidance to states to which conditions they should be doing newborn screening for. And for those of you that aren't familiar, newborn screening is simply taking a blood spot, drying it on a card, and then being able to send that to a reference lab and have them determine enzymatic activity for a whole panel of enzymes. Again, looking for ones that would be abnormal and potentially identifying patients that would benefit from early treatment of these therapies. So here's the status of newborn screening for MPS I. The majority of states are now screening for it. Began in 2015 formally in Illinois, and now you can see where it spread. With this much newborn screening and testing, this is a good example of where we're generating a lot of uncertain variants, and we need these functional assays. So as all these conditions get added to the recommended uniform screening panel, you have a lot of states testing for it. If you get abnormal screens, typically what's done right after that is you sequence the gene where you think you have an abnormal. When you sequence the gene, you identify variants, and a lot of those variants, like a lot of conditions, are uncertain significance. So the functional evidence that would help support this effort would not only help determine pathogenicity and decide whether you should treat or not, it also adds experimental evidence that can be used by these expert curation panels that could then formally reclassify the variants of uncertain significance to benign or pathogenic. And then, as I mentioned before, the tools from these functional experiments and outcomes can be used to test or identify new therapies. So I got into this probably 3 or 4 years ago when a few of my colleagues here at Greenwood were specifically interested in having someone develop a platform for IDUA variant classification. And I had really always enjoyed working on lysosomal enzymes from my time in Stuart Kornfeld's lab as a postdoc. And so I said this would be an interesting challenge, and we took this on. This is how the platform works. So we start for all of these platforms with HEC293 cells that are knocked out of the gene of interest, in this case, IDUA. We transfect in either wild type or variant IDUA expressing vectors, and then we'll typically do an antibiotic selection to remove all the cells that didn't get transfected. If we need to, we can proceed with single cell cloning. That will give us single clonal cells that overexpress the different variants. This is helpful if we need to find a clone that has a very high level of expression when we're dealing with an enzyme that has very low activity. But essentially, for all these platforms, we can go from any one of these levels and move on to the analysis. And what we do is two very simple assays. We measure activity using fluorescent substrates, in this case for MUI, and we measure that relative to wild type in protein normalized lysates. And then we do a semi-quantitative determination of iduronidase levels. And we do this by Western blotting. And I'll show you some examples of that. But essentially, we get an activity relative to wild type and an abundance or amount relative to wild type. We put these two factors together to give us what we call a relative specific activity, or an RSA. Very simple terms, it's just activity over amount. We believe this has a lot of benefits from just determining activity alone, because again, you have to ask yourself, do you have an unstable protein? Is that what's causing the loss of activity? And I'll show you in a minute, we learn a lot of other things from doing the Western blots. But this also gives you a single value that you can use to basically assign each variant, line these up, and then ask questions about where's the line between potentially the pathogenic and the benign variants. And maybe not to anyone's surprise, this determination is not that easy to just draw a line through. But we've done probably 100 of these now. I'm only showing you a subset. And this is just the RSA values of these individual variants lined up. We included several known pathogenic variants. Those are in these colors here. You can see where these fall on this chart. The vast majority of them are below this line. This is on a log scale, so this is 1% of wild type relative specific activity. A lot of these fall below that line, and many significantly, several orders of magnitude below. We included benign polymorphic changes. These are ones that have a very high allele frequency within the general population. They're definitely not disease causing. And these, group over here where you have wild type. The reason to include both known benign and pathogenic variants in this type of analysis is you need to be able to have your platform powered sufficiently for then these expert curation panels to come in and confidently use the results that you get to reclassify the variant. So it's just sort of a necessity for building a platform that could be used in that purpose. But we'll touch on a few of these things. What I can tell you is the story isn't that simple. We do know there are some pathogenic variants that fall above some of our known benign variants. And in the talk I gave last week, we sort of addressed issues around needing to do just other assays beyond just this simple RSA determination in order to fully appreciate what's happening with these variants in the context of the cell. But let me just tell you a little bit more about the Western blotting. So when we look at this, the Western blots, we get an abundance, a total amount of the protein that's there, but we also get some idea of its maturation and processing. And that's because alpha iduronidase, like a lot of lysosomal enzymes, when it reaches the lysosomal compartment, gets chewed back by the proteases that are there into these characteristic bands. For iduronidase in the wild type case, we typically see three major bands. You'll see those on other ones. They just represent forms of the enzyme where we've trimmed back either from the N terminus or the C terminus. We know this trimming is lysosomal because if we block lysosomal function, like with baflomycin, we end up shifting this all back up to one of these precursor forms. So it's a good indication of whether the enzyme has actually reached the lysosomal compartment if you either see processing or you don't see processing. It's basically an indirect measure of lysosomal trafficking. Here's what the data looks like. You can see there are some that don't appear to process at all. Here's the wild type, these very nice three characteristic bands with that middle one being the major one. And then some that clearly do process, some that weakly process. Again, this is giving us an idea of whether it's maybe reached the lysosomal compartment and what its behavior is, and just basically how its maturation is affected by the variants. All right, so now I want to give you just a brief little other advantage of this platform before we jump into the glyco side of the story. And if I go back to this picture here really could spawn probably half a dozen different stories. And I don't have time to tell you all of them, but I'm going to tell a couple of them, one of them before we get to it. So one other situation that people are running into when they do the newborn screening is they're getting genotypes that don't just have two variants. In other words, one on each copy of the gene. They're getting these situations where they might identify two what appear to be clinically benign variants on one allele, and then a very obvious pathogenic variant on the other allele. This opens up the question. Now, normally, if you just had a single clinical benign variant and then an affected, you wouldn't expect that patient to be affected. But now you have this additional variable of what if you're dealing with two variants? Is there some combination effect? And this turns out to show up more frequently than I think most people appreciate. In fact, I was in touch with a group in Canada that said they had a case where there were 7 IDUA variants that were identified. So you can imagine the combinatorial possibilities with something like that. But this is what some of these look like, right? You're picking up 3 different mutations. Sometimes you know the phase, they're in cis. This would mean that they're on the same allele, and then versus trans being on different copies of the gene. Sometimes you don't know the phase, but this led us to ask, can we provide some functional insight into this scenario? And we can, because what we can do with our platform is not just express a single variant enzyme, but we can include one or more IDUA variants and then functionally test the impact of that. And so what we learned, and this was reported last year, is kind of interesting. So all of the in-cis combinations where we have 2 variants on one copy all resulted in a relative specific activity that was lower than the individual ones. So you can see these different combinations here. This one has pretty good activity, V573L. This one's a little bit lower, but when we combine them, we get even lower. And then other examples like this one, G409R and A79T, both well-known benign variants, but you combine those, where is that one? And you get down again, getting closer to this 1% line. So all of those examples, I think, alerted us to say that if you have 2 or potentially more variants that you'd normally consider to be clinically benign in combination, you can start to get into a range where you pick up other attenuated variants right around that 1% line. And so I think the field is starting to pay a little more attention to this issue. And one other really interesting thing that we found is that several of these combinations that we made combined what would normally produce an enzyme that's not very well processed and one that is processed. And in all of those cases, like here, these two, G409R processes beautifully, just like wild type A79T does not. When you combine these, you basically block processing. Same thing with these ones over here, right? R435G is a non-processor. This V573L is a processor. You combine them, you don't get processing. So the non-processor is kind of exerting an interesting dominant effect on this. But I just wanted to share that as one side story before we move on to sort of the next major point that has stemmed from this initial work. And this got started when we were considering the very large difference in relative specific activity of two alanine to threonine changes, one at position 75 and one at position 79. And this was surprising because these are on the same alpha helix, about one turn away, but there was, over 1,200-fold difference, lower activity for A75T compared to A79T. So what was the basis for this? Well, we took a closer look at the sequence, and what we found, quite surprising, is that the A75T change actually creates a new consensus sequence for N-glycosylation. So here you had NLA, now you have NLT. That again fits with the consensus sequence, which would be asparagine, any amino acid besides proline, and then a serine or a threonine. So we predicted that this would create a new sequon, and then an extra N-glycan should be added to the enzyme, which would increase its apparent molecular weight if we were to do a Western blot. And that's exactly what we found. So here's the wild type. Here's G84A. This is just a non-processor. And then here's A75T. And I think you can nicely appreciate that shift higher, about 2 to 3 kilodaltons. That, those of us in this field always appreciate when you see a shift like that it could be the addition or the loss of an N-glycosylation site. So that kind of lines that up a little bit better. Remarkably, this variant has been appreciated to be a pathogenic disease-causing MPS1 variant for probably a decade and a half. No one understood the mechanism, and no one really appreciated that variant was actually creating a gain-of-glycosylation site. So a joke I always make is this is why you have to have a glycobiologist in every field so they can notice these things. So this was another one that had been identified in patients, P183S. This one doesn't really look like it shifted, but we did some interesting experiments just to kind of gauge the nature of the sequon. We made the corresponding, for A75T, we made A75S. For P183S, we made P183T. And it turns out that when you have A75S, you don't get the shift. When you have P183T, you do get a shift, whereas you didn't with the other versions. And this corresponds with effects on activity as well. So here you could see A75T, very, very low activity, essentially dead. But if you make the S and you don't add that extra N-glycan, you have decent activity. It's not wild type, but it's certainly better than this version. And the same thing holds for here. When you add an extra N-glycan, it's correlating with a very strong effect on activity. Whereas when you don't, those effects are not as dramatic. So in order to confirm that we were actually adding an extra N-glycan, we worked with Merriam and Peristu at the CCRC to do glycoproteomic analysis on wild type and A75T expressing cells. And what they were able to show is for these, we were seeing addition of an extra N-glycan at N73. So that's the asparagine that will get modified when you have A75T, whereas we weren't seeing that in wild type. One other interesting thing came out though. While you had a diversity of different glycan structures for the wild type, including some with fucosylated, apparently complex structures, and those that had trimmed high mannose, which would be indicative of glycans that got mannose 6-phosphate added to them and then were trimmed back in the lysosome. The only thing we found on A75T is all untrimmed high mannose type glycans. So this would support that when you add that extra N-glycan, you're essentially trapping this enzyme, likely misfolded, in the ER, and that's the primary mechanism of why you have such low activity. To look at this a different way, we have the extra N-glycan being added to the enzyme. It's causing misfolding of the enzyme, and you're not getting it to go to the lysosome. So this is supported by, again, having very low activity and no secreted activity at all, and then all the N-glycans being found in unprocessed forms. Okay, so we took a look in ClinVar, which I think maybe many of you are familiar with. It's really just a database where people that are identifying variants in genes can deposit them into a common repository. It's a wonderful database. It's searchable. But we asked what other IDUA variants that have been deposited in ClinVar would have a predicted gain of glycosylation effect. I've already talked about these top two. I'm going to show you some data in a minute on these last four. But one thing you can appreciate that's also in ClinVar is a classification. And this is just the formal classification that's used by clinicians to determine, what the effect of the variant is. Well, this was well appreciated, A75T being pathogenic, but all these other ones were VUS. So a lot of times when people are running into variants that they may not even realize have a predicted gain of glycosylation effect, these are of uncertain significance. Now, what we did is we went on and looked in ClinVar at 8 other MPS disorders and found 35 predicted gain of glycosylation variants. And I'm just going to poke through these in the next couple slides so you can appreciate the ones that we found. One thing that will emerge, you'll see again, there are a number that are still variants of uncertain significance. And all these ones I'm going to show you, we are planning to do functional analysis on to confirm their effects and ask whether they do truly lead to a gain of glycosylation. But this would be a good point of discussion at the end as to how maybe we could first do some work in silico to sort of help us from having to do all the bench work. You can tell some of these, like this one for MPS2, is pathogenic. Again, our prediction now would be that this definitely adds an extra N-glycan and likely leads to misfolding of the enzyme. Some of these had multiple, others we just found single ones. And then here's the remainder of the list. So MPS3D, this has an interesting one at this site, 185. You can either get a loss of glycosylation if you delete this asparagine, or gain if you add this one back in. So some of these regions are just really complex. And then some of these I was surprised at just the number. So for MPS 4A, a huge number of ones that have, and several that are predicted to be pathogenic, have already been identified. So this just gives you an idea of what's ahead. I don't think we want to stop at just the MPS disorders. There are probably 30 or 40 other lysosomal storage disorders that would be caused by soluble hydrolases. I think it would be very interesting to expand this. All of this work identifying these variants was actually done by my nephew, who was a high school student in North Carolina. He did this manually going through ClinVar. I would love to have a discussion as to how we could mine these in a more efficient way. And I would be surprised if there isn't someone on the call who says, yes, that's easy to do. In addition to the gain of glycosylation ones, we also looked for ones that would cause a loss of N-glycosylation on IDUA. And you can see this list down here. So there are a number. Almost all of these are considered uncertain significance. We don't really know what the functional impact of loss of N-glycosylation would be. Certainly, if you're altering glycans that bear mannose-6-phosphate, you could imagine impacting the lysosomal trafficking, although there are usually multiple N-glycans that are modified. But some of these might just be in a region where they have unexpected functional effects on the enzyme. So I think eventually we'll turn our attention to some of these loss of function variants as well. And even here, so this is MPS2. You can see again a whole number. And again, the loss could occur due to changes in the asparagine or changes to the serine or the threonine within the sequon. And my nephew Ben even found interesting examples, right? Here's a special case where it's not a loss of one of those key amino acids, it's a change from a threonine to a serine. And is that enough to change, is that enough to change the utilization at a different sequence? So again, we're dealing only with the ones that have been deposited. We're not sampling the entire space of what's possible in terms of sequence changes, only the ones that have been identified. Let me jump back to those other three. So here was something interesting that I think is, again, could be built out further. These are the other three IDUA variants that we studied. And one thing that we noted is that all three of these look like they do have the extra N-glycan added. So for these two, D243N and D44N, you can clearly appreciate that shift by looking at this middle band. But even this one, N350S, is shifted up from this top band here. So this band across the very top here is just a non-specific band that we look at. Now, we thought that addition of the extra N-glycan would correlate with loss of activity, but only one of these, N350S, had this significant decrease in activity like we noted with A75T. The other two had a very modest effect. In fact, this one barely at all. So what is the reason behind that? Well, that came from looking at the structures and looking at where the extra N-glycan would be added within the folded enzyme structure. And for this, we had the help of Kelly Mormon at the CCRC, who did AlphaFold 3 predictions with these extra N-glycans in place. And I think you can appreciate this very easily, that these two that had a very strong effect on activity put the extra N-glycan into the middle of the folded structure, and this creates strain. This basically should never fold, as I understand from Kelly, and you can see that in this sort of lighter color that you've got real steric hindrance when you're adding this. In contrast, look at these other two. These are now putting the extra N-glycan on solvent exposed loops on the surface. So they're probably not affecting the folding, and they're just sort of there on the surface with, from at least the ones we've studied so far, a very modest effect on activity. All right, so let me move on to this part about therapy, because when we started to appreciate that these extra glycosylation sites being occupied, or this gain of glycosylation was causing a damaging effect on the enzyme, it opened up the possibility that we could use inhibition of N-glycosylation as a possible treatment avenue for these variants. Again, this would only work for specific amenable variants, and it has to be somewhat selective in nature, because we don't want to start stripping away all the N-glycans on this enzyme. It needs them for folding. It needs them for protection in the lysosome. In the case of iduronidase, there's a specific glycan that's actually involved in substrate binding. So it has a more direct role in catalytic function. So how could we do this selectively? Well, in the past, most of the N-glycosylation inhibitors were at the level of limiting the amount of lipid-linked oligosaccharide precursors needed for N-glycosylation. That's not going to be very selective. So thankfully, a few years back, a colleague and friend, Joe Contessa, set out to find new N-glycosylation inhibitors. He was doing this because he was interested in inhibiting N-glycosylation on receptor tyrosine kinases, that are needed for the growth of tumor cells. So he's basically trying to find an N-glycosylation inhibitor that would serve as a cancer chemotherapeutic. And he came across this compound, NGI1, that you can see from that Western blot, inhibits N-glycosylation occupancy in a dose-dependent fashion. And what Joe went on to discover is that NGI1 actually acts at the level of the oligosaccharyl transferates. It is a somewhat redundant but selective inhibitor of the two catalytic subunits of that complex, STT3A and B. And by doing this, you still have the same amount of N-glycosylation precursors, but you're just blocking the addition at different sites. Again, depending on whether you're inhibiting, the A subunit or the B subunit. And I won't go into the specificities of those two subunits in this talk. But we had the tools. So we had our cell line that expressed A75T, and we treated with NGI1, just asking if we treat with this glycosylation inhibitor, do we now start to see activity? And we started by just asking, do we see secreted activity? Remember, this is an enzyme that is misfolded sitting in the ER. But what we found is that if you treat with in different doses of NGI1, you start to now see secreted iduronidase activity, even more so if you treat for a longer time period. This was exciting. This meant that we were restoring some iduronidase activity by treating with this inhibitor. If we increase the dose, you see that effect start to wane. And we think that's because increasing amounts are going to hit more and more glycosylation sites. You can appreciate that from this Western blot. And it also starts to affect stability, probably because you're losing N-glycosylation sites that are needed to stabilize the enzyme in its eventual compartment, the lysosome. So that's just maybe a potential limitation with this. But if we look at not only the secreted activity, the intracellular activity after treating with NGI, what we can appreciate is that while we had very low, this is at the relative specific activity, again, well below, into the pathogenic range. But if you treat with increasing amounts of NGI1, you start to get back up towards that 1% level, which is kind of, we view as the attenuated range. So it's possible that these drugs could be used to take a patient with a severe mutation like A75T back up to an enzyme level that's closer to an attenuated or more mildly affected patient. So Joe has a number of inhibitors that he's working with and has developed. Some of these are specific for different OST subunits like STT3B or A. What we did is treat these same cells with these different inhibitors, and you do see some interesting selectivity. So this one, 624, that like NGI1 is kind of an inhibitor of both, works even better than NGI1. So I think where we're going with this is, we have a number of these predicted gain-of-glycosylation ones. We need to confirm that. And then we have this whole other layer of asking which ones are amenable to which of these growing number of inhibitors. And so I think that's kind of the work that's in front of us. But the basic idea here is that you have the misfolded enzyme in the ER. If you treat with these inhibitors, you block the addition of that extra N-glycan on a subset of the enzyme. This allows it to fold, be trafficked to the lysosome, and secreted outside the cell. So we were still trying to tackle some site mapping studies to show that NGI1 does in fact block that extra N-glycan. It's just can be challenging to work with an enzyme that's already kind of misfolded. All right. This is the slide I was referring to that hopefully is a jumping point for further discussion. So some ideas and things to think about. What types of databases or resources are needed for the medical genetics and glycoscience community that stems from this work? What does the field need to see? What do both fields need to see? Are there better bioinformatic tools to predict the gain and loss of function variants in ClinVar? Is there an automated way to say, take different genes, start with all the lysosomal storage disorders, and give a comprehensive list of what's there. What a great jumping point project for people to say, okay, I'm going to see what these look like. And then I touched on this. We thought this was incredibly valuable, what Kelly did for us, but how to couple the structural biology of the enzymes. And a lot of them have, solved crystal structures. If not, again, there are programs like AlphaFold 3 into the predictions for variant effect. My assumption is that the majority of those that are currently listed as pathogenic probably put that extra N-glycan. They do add it, they put it in the middle of the structure, it blocks the folding, and you basically have an enzyme sitting in the ER. And then how do we catalog amenability for the different variants to this growing collection of these OST-based inhibitors? Joe is moving some of these forward into the clinic. My understanding is some of these look really good. They're shrinking tumors in mice. They may be compounds that are clinically available and could be repurposed for the treatment of some forms of MPS 1. So with that, I'll stop. I have a whole list of people here to thank. In particular, staff scientists in my lab, Sako Yu, who has really done all of this work, but it's been supported by wonderful colleagues and collaborators at the CCRC and elsewhere, and supported by family foundations, including the National MPS Society and Cure San Filippo. We have platforms now for MPS 2, and we're working on MPS 3A and B. So we'll hopefully have the whole tool set to work through these as some of these conditions are added to the recommended screening panel and newborn screening is done. So look forward to your questions.